Hello, everyone. Welcome to Penn State CTSI Bird Seminar Series in this fall. This seminar series is co-hosted by me, and my name is Jay Zhu, and also by Dr. Lan Kong. We are both faculty members in the Department of Public Health Sciences at Penn State. This seminar is set up as a Zoom webinar. I will just go through some basic logistics for this Zoom webinar. This Zoom session is being recorded automatically. Then you will get a video link afterwards. All the attendees are muted except for the panelists. If you have any question, please feel free to submit your question using the Q&A box. If you are registered attending, you will actually receive an email with the slides and the recording link. And also there's a seminar feedback survey afterwards. We do encourage you to register for future seminars. You can go to ctsi.psu.edu and then go to research support and then seminars to register. Today, we're very pleased to have two of my colleagues, Ms. Kelly Nye and Dr. Paula Valencia-Moulton here. Kelly is a research professional with more than 20 years of experience at the Penn State Department of Public Health Sciences. She is the project lead for the clinical trial monitoring services. Since joining Penn State, she has conducted data management, project coordination, and clinical trial monitoring services for single and multi-site clinical trial research. Kelly is a certified clinical research associate with the Association of Clinical Research Professionals, ACRP, since April 2022. Dr. Valencia-Moulton is a senior research project manager at the Penn State College of Medicine. She has over 15 years of experience developing data management infrastructure and utilizing innovative technologies for data collection and quality assurance of clinical research data. Since 2017, she has been leading the data management and the clinical trial monitoring teams within the Department of Public Health Sciences. The topic of their lecture today is clinical trial monitoring. Ms. Nye will be the primary presenter. Welcome, Kelly and Paula. I will let you get started from here. Let me stop sharing my screen, and you can go ahead, Kelly.
Okay, thank you for that introduction. And we'll go ahead and get started. So today we're going to be talking about clinical trial monitoring, and regulatory agencies and research experts from pharmaceutical industry, academia, and government agencies promote the use of risk-based monitoring components in a risk-based quality management plan for clinical trials. So today we're going to talk through proactive risk-based monitoring components and how they can be utilized in your clinical trial monitoring plans. So we'll start just by going over the purpose of monitoring, and this is taken from the International Council for Harmonization, or the Good Clinical Practice Guidelines. And the purpose of monitoring is to verify the rights and well-being of human subjects are protected, the reported trial data are accurate, complete, and verifiable from source documents, and the conduct of the trial is in compliance with the currently approved protocol and amendments, good clinical practice, and applicable regulatory requirements. So risk-based quality management is not new to clinical research. In 2005, the ICH produced a Quality Guideline 9 for quality risk management, and this provided an overview of a typical quality risk management process. This followed in 2008 to 2010, the Clinical Trials Transformation Initiative had a quality objectives of monitoring project. And in this project, they reviewed the current practices of monitoring, the key quality objectives of monitoring, and the qualitative assessment of monitoring techniques. And there were representatives from a variety of organizations involved in clinical research, and they were asked to share their perspectives on the key objectives of monitoring. And they identified patient safety, accurate representation of key data, protocol compliance, and quality improvement as their top 4 objectives. They also indicated that monitoring is most useful when it identifies errors early so that corrective training can be carried out. Another theme that came out of this project was that the regulatory agencies should provide more specific guidance related to their expectations for clinical trial monitoring. So in 2013, we see this regulatory agency documentation and experts coming through with this guidance. The European Medical Agency's reflection paper on risk-based management is a key document. Also, the FDA oversight of clinical investigations, a risk-based monitoring approach. They all promote the use of risk-based monitoring in clinical trial management. Also, the Transcelerate by Pharma Incorporate position paper on risk-based monitoring is another in-depth look at risk-based monitoring methodology and is also highly cited in the research. In 2016, the ICH produced an integrated addendum to their good clinical practice. So that was the revision 2 for those guidelines. And they continued to add more risk-based quality management items to the guidance documents. In 2018, the FDA formally incorporated the good clinical practice guidelines into their guidance for industry. And in 2021, we see there's another revision of the ICH Good Clinical Practice Guidelines, and a draft principles document is issued to give everyone an idea of the changes that are going to be added. And they do highlight quality by design, risk assessments, and a multidisciplinary approach to be involved in the design and implementation of the clinical trial. So the traditional monitoring method is a frequency-based method, which traditionally starts at the first enrollment in the clinical trial, and then the visits are based off of a certain frequency, either a time frame, 6 to 8 weeks, or a frequency of enrollments in the study. And they're typically on-site visits, and they're uniform across all sites. The on-site visits focus heavily on source data verification, which is a transcripting checking process. And it's a very reactive approach to the problems and very resource intensive. So in comparison, the risk-based approach begins with a risk assessment, and this happens prior to the first enrollment of the study, where system level and project level risks are analyzed for the likelihood and consequence of their occurrence. And then based on that risk assessment, risks are either mitigated or accepted. And there are several different approaches to monitoring that can be taken to incorporate into the monitoring plan. And those are off-site remote visits, central monitoring, targeted on-site visits, a reduction in source data verification, and in total, this is a more proactive approach to prevent risks from occurring and is a more strategic use of resources. So I'd like to pause here and get a sense of your experience with risk-based monitoring. If we can launch the poll, and we'll give everybody 10 seconds or so to answer this question. So how familiar are you with risk-based monitoring components? Very familiar, somewhat familiar, not familiar at all, or not very familiar?
I think most of the participants answered, so
Okay, we can go ahead and end the poll then.
yeah. And just share the results.
So there is some familiarity with the concepts, and that's good to hear. And hopefully you'll be able to share some of your experiences during this presentation as well. So an example or a snapshot of how risk-based monitoring is being used in the clinical research setting. The Association of Clinical Research Organizations represents a clinical research and technology companies that provide a variety of specialized services to support the development of new pharmaceuticals, biologics, and medical devices. And they did a landscape survey in 2019 and 2020. And this just serves as one example or a snapshot of how risk-based monitoring is being used. In 2019, their survey was conducted and over 6,000 ongoing studies were involved, over 7 contract research organizations, and 47% were using at least one risk-based component, and 53 were still using the traditional approach. In 2020, there was a little less, almost 6,000 ongoing studies, and there were 908 new starts in 2020, overlapping 6 contract research organizations. And you can see there is an increase in risk-based monitoring approach, 77%, and 23 are still using the traditional approach. And this is important because the pandemic was going on in the 2020, so you can see that that might have spurred more risk-based monitoring approaches. This next slide shows figure 3 from the ACRO risk-based monitoring and clinical trials increase adoption throughout 2020 paper. And the authors noted, quote, the landscape survey data shows year-over-year growth in risk-based monitoring and risk-based quality management component implementation, but adoption rates overall among all ongoing studies for the individual components and the risk-based monitoring, risk-based quality management frameworks are still low. The 2020 new study start data shows a sustained increase in the risk-based monitoring adoption. So this is, again, just another snapshot of one group that is using risk-based management in their clinical trials, and it's relevant and recent in 2019 and 2020. So obviously not everyone is transitioned to the risk-based monitoring approach, and we want to find out why that transition is not occurring. The regulatory guidance have been out for a relatively long period of time, coming upon 10 years now. So the regulatory guidance is there to support the process, but there may not be awareness and education down to the clinical trial management teams. So that's one thing we'd like to increase the awareness and education for these monitoring methods. There may be some misconceptions that this process would not fit all studies, and the reduced source document verification may lead to more audit findings. There's also a complexity of implementation of these methods. There are logistical barriers. Data often is coming from multiple data sources, and all of those data sources need to be combined into one electronic data capture system or a data set to perform the central monitoring functions. And the data may not be, and there's a lot of systems that are being used. The research teams have to have logins and passwords to remember for those, and training on all of those electronic systems. So those may be some logistical barriers. Also, the allocation and resource time for the clinical trial team to upload source documents to a remote system for remote review and training on that software. I think the COVID pandemic has kind of forced that to happen a little bit faster. So that is one good thing that has occurred. And there may be a reluctance to changing existing practices and procedures. And this really does need to be a multidisciplinary approach. And the research teams would need to practice these risk-based monitoring practices to see the results and make them more comfortable with the changes in procedures. So now we'll take some time to go through each of the risk-based monitoring components in more detail and provide examples. So the first is the risk assessment, and this starts prior to the enrollment. And the team wants to identify all of the problems or risks they can see at the system level and the project level. They want to determine what might go wrong, what is the likelihood that it will go wrong, and what are the consequences if it does go wrong. And that's in the risk assessment portion of the diagram. Then they'll move into the risk control, where they'll determine whether the risk needs to be reduced or mitigated, or if they will accept that risk occurring. All of the risk assessment should be documented in a risk management plan to indicate the mitigation strategies that will be used in the clinical trial. And the risk management plan should be reviewed when risks occur, and also when the protocol changes would initiate another review of the risk assessment process. And the two pillars on the side of the risk assessment process identify communication and the use of risk management tools to help with this process. And this risk assessment is from the Quality Risk Management Guideline from the International Conference of Harmonization. So this is a basic example of risk assessment where you would identify the risk clearly, and the study team would then assign a likelihood score and a consequence. Those are multiplied to get a total risk score, and then those risks are assigned to a category. In this example, the categories are just listed as A, B, or C. That can be changed if the study team would want to use colors to identify risks or a high, medium, low process. There's also an Excel format of the risk assessment that I can share. And this allows the study team to sort and filter their risks and organize them by categories. This first column identifies whether it's a system level risk or a project level risk. The next is a subcategory, and the study team can adjust those categories based on the protocol, identify the risk, and then assign the likelihood and consequence scores. And when those are updated, the risk score auto calculates and the category is updated. So it just adds a little bit of functionality there. The control decision is documented either to accept the risk or mitigate. And then if mitigation is decided, then you would document the mitigation strategies that would be used for that risk. So in public health sciences, when we start our risk assessment review, we're starting with the review of the study documents and the electronic data capture system. From the project level, we're looking for critical values being captured in the study documents and in the EDC system. We're looking for the secondary endpoints. Sometimes we see that those are missing, or laboratory values that are used to verify eligibility criteria. So those are the things that we're making sure that are in the EDC system before the study would start. Also documenting the final disposition of each enrolled participant, the study drug compliance for an overall metric at the end of the study. And also one common thing that we find is the order of ethnicity and race that is collected in the EDC system. There's FDA guidance documents on the collection of race and ethnicity in clinical trials, and it specifies that how ethnicity and race should be collected, and ethnicity should always be collected prior to race. So we always make our teams aware of that. We also have a working knowledge of REDCap, and a lot of our teams use REDCap to store their data. So we do have experience working with randomizations and helping the research teams set up the randomization, test the randomization, and also make sure there's a plan for if REDCap is down, they have a backup method to perform that randomization. Survey distribution, we will look at the automated survey invitations to make sure the conditions are set up according to the protocol and they will be sent as expected. And also that there is a control in that process to stop the invitations if the participant would leave the study early. Other mitigation strategies that we use is our best practices for electronic instrument design. And this may include setting required fields, marking identifiers in the EDC system so that they're not accidentally exported, establishing validation to control data entry and data quality control. We also highlight tools and resources posted on the College of Medicine Clinical Trial Office and also within the Clinical Research Guidebook. And we have developed a REDCap regulatory binder to support the regulatory document storage for teams that are using REDCap so that your study data and your regulatory data can be in the same system. And this is a template that any Penn State researcher can use for their study to keep their regulatory documents organized. And we'll take a deeper dive into the e-regulatory binder later in this presentation. There are two other risk control processes that are not as heavily used, but they're very important, and quality tolerance limits were defined by the European Medicines Agency as the initial acceptable variation or tolerance limits for the clinical trial data or procedural metrics. And these really need to be tailored to the protocol and the critical data for the protocol. So that's where you want to tie in those quality tolerance limits. And they can be applied to trial data, procedures, good clinical practice, or just overall trial management procedures. Key risk indicators are another terminology that's used, and they differentiate between the quality tolerance limits because they're defined to differentiate general or site metrics from the protocol metrics. So some examples of quality tolerance limits and key risk indicators. It's a little bit hard to give you examples of quality tolerance limits because they are so protocol specific, but one that I see frequently are applying visit windows to study encounters, and this helps for scheduling visits. It gives a level of tolerance for the scheduling, be it plus or minus 3 days, 5 days, 7 days, but it does give a tolerance limits for those. And if the visit would happen to be scheduled outside of those limits, then at that point, it would be considered a deviation and tracked in the research records as a deviation. Also, deviations can be a quality tolerance limit. the frequency or distribution of deviations across sites could be another tolerance limit example. Key risk indicators, there could be a change in PI or a key study team member change, and this could initiate a remote review of the delegation log and the study training to make sure the delegation and study team members are qualified for those duties before they would start working on the study. The rate of AEs per participant could be another key risk indicator that could trigger a remote or on-site source document review to verify those AEs that are entered. The number of AEs or the number of days between visit date and entry could be another trigger to contact the site and remedy the late data entry and make sure that the data is being entered in a timely fashion. Days since last screening visit could also trigger a contact with the site and the site PI to determine if there's barriers to screening and recruitment at that site to make sure the recruitment stays on track for the study. This leads us into central monitoring, which again is a remote data review in the fact that it is performed by staff members who are not involved in collecting the data. It's performed by the sponsor or a representative and can be conducted by a monitor, a data manager, statistician, or a safety monitor. It would depend on the type of review, the expertise that is required for that review, on who would complete that. It's looking for unusual data patterns. Data checks are often used to look for missing or invalid data, and compliance data metrics are also reviewed during central monitoring. To take a deeper dive into central monitoring, we'll look at some data quality rule examples. So REDCap allows data quality rules to be executed real time during data entry. This is an example of a data quality rule that would come up for an adverse event update. So the clinical research team has added a stop date to an AE, and the outcome is still marked as still present. So this would flag the error for the research coordinator to investigate. They would verify that they're in the right AE to add the stop date. And if that's correct, they would then look at the outcome to verify if that needs to be updated for this adverse event. And they could correct this at the time of data entry, and then that would be resolved at that point. REDCap also allows for data quality rules to be executed on demand within REDCap and queries issued by a data manager or a research coordinator. Also, the data can be exported from REDCap to use in other systems such as SAS or Tableau. This is an example of a SAS data quality rule report where a prohibited medication was entered on a concomitant medication form. In this example, the data manager would investigate the entry in REDCap and issue a query to the site staff to confirm that the entry is correct and take further action as necessary. And data quality or data exports from REDCap can also be on demand manually, or it can be an automated process. Another central monitoring component is the use of data quality reports, looking at enrollment statistics, missed visits, or number of queries issued unresolved or closed. Those could all trigger actions to be taken. Study accrual reports monitor the number consented, screen failures, enrollment, post-enrollment termination, and completed participants. It can also include a breakdown of screen failures or post-enrollment terminations if there is a indication there for further action. And the date of last screening or enrollment visit can sometimes be used to indicate enrollment or screening issues. For central monitoring, there's also the use of trial management and subject tools. A trial management tool example is the e-regulatory binder, which Public Health Sciences Data Management Unit updated from a template that was shared from the Partners Human Research Quality Improvement Program and Enterprise Research Infrastructure Services in affiliation with Harvard Catalyst. So they were able to share their template with us, and we were able to update it to be a multi-site or a single site regulatory repository and share that with the Penn State investigators for use within REDCap. REDCap e-regulatory binder uses repeating instruments to display information at a glance. This is an example of 3 repeating instruments where you can see at a glance how many IRB reviews have occurred, how many protocol revisions, and staff documents, just as an example. Reports can also be generated to generate IRB submission logs, protocol or consent amendment logs. This is an example of a consent tracking log. So once the consent information is entered into REDCap, it can be reported back so you can see at a glance for a particular study all of the consent versions and their date of IRB, valid date, and expiration date, and a reason for the change in consent. Also, alerts can be programmed into the e-regulatory binder, and this is helpful for IRB submissions when an IRB determination letter has been received and we need to share that with the study sponsor. An alert can be programmed to be sent to the regulatory associate, and this would be an example of an alert that they would receive telling them that there is a new submission. They would go into the e-regulatory binder, download that, and send that off to the study sponsor so that we stay in timely compliance with that. Another subject tool for central monitoring is the visit schedule, and that can be generated from the electronic data capture system or use of an Excel spreadsheet tool. And I can show an example of the Excel spreadsheet. So you can see it can be customized for the study to break it apart into screening and baseline schedule and a post-randomization schedule. And it's helpful to present this at the time of consent to the participants, letting them know what's involved in the study, how long it's going to be. It's a great visual for them to see if it's something that they can really commit to and see through. Also, it can help when scheduling. A lot of clinical research staff try to schedule early in the window so they have more flexibility to reschedule if life events happen, and also for the participant to identify future events that are coming up that they may need to work with a research team to make adjustments to their scheduled visit. Offsite and remote monitoring are typically conducted by monitors, and an example would be a remote access to the EMR to conduct a targeted source document review. An example for this could be that example of a high frequency of AEs for a particular participant. The monitor could go into the EMR to look at that source document review and verify that data. A targeted remote source document verification, again, is that transcription process, could be identified from central monitoring when there's a high rate of data entry changes, and a remote source document visit could be scheduled to verify those changes and also work with the study team to decrease that rate of data changes if it is a data entry issue. Also, offsite remote monitoring can be done on the e-regulatory binder if it's electronic, and review can be triggered when a key change in staff occurs. The monitor can go in and look at the delegation log and the study training documents. So source data verification should be able to be reduced in a risk-based quality management plan and also allow for an adequate use of resources. Examples of this reduction would be a targeted central monitoring to identify key risk indicators of data changes, reducing source data verification to only the form that captures the primary endpoint, or conducting source data verification on an instrument where there's a high risk of error rate. And this could be a laboratory results instrument where the data is being transcribed from a report to a paper case report form. And then that paper case report form is then entered into the EDC system. So there's various levels of error that could occur. So there is evidence in the literature indicating that source data verification may not be the best tool to ensure the data quality of the study-wide. One example is from the Transcelerate by Pharma position paper on risk-based monitoring methodology. It had a 9-sample study from 6 member companies, and they looked at the source data verification generated queries. Overall, the rate of those queries were low, and the average percentage of source data verification queries generated was 7.8% of the total number of queries generated. So there were a lot of queries coming from that central monitoring process. And the average percentage of the critical data queries that were represented from the source data verification was only 2.4%. Another example comes from the evaluating source data verification as a quality control measure in clinical trials paper, where they did a multi-study data analysis in partnership with Metadata Solutions to look at the impact of source data verification. Over 1,000 phase 1 through 4 studies were used, 53 sponsors, and a median of 1.1% total electronic case report form data set was corrected by source data verification. Audit findings are another example from Transcelerate. And this is member company audits from January 2012 through May of 2014, 8 companies. They included critical and major findings. Each of those findings were categorized of how they were reviewed. And the results were over 1,000 findings. Only 11% were related to source data verification, and 0.65% are related to critical findings. So there's definitely evidence that relying on 100% source data verification is not the best way to ensure overall quality of the trial. And we need to target that process based on other risk-based monitoring components. So source document review is a little bit different than source data verification, where we're looking at the quality of the source and reviewing it for protocol compliance, making sure it's adequate and the investigator involvement is documented and there's appropriate delegation to tasks. So an example would be the regulatory document review at pre-enrollment. We're looking at study tasks that have been delegated to research staff. We're looking at their training and experience and making sure that aligns and that protocol training is documented and complete. Another example of delegation of tasks would be the physician review and evaluation of out-of-range laboratory values during the study. At source document review, you would be able to see that documentation from the physician and make sure that that is occurring as delegated for the study. So in a wrap up, monitoring plans should be trial specific. It's not a one size fits all. They're created by the sponsor and the clinical trial monitoring group. It should be developed based on a risk assessment and a management plan. Include a variety of monitoring activities. It should be reviewed with the study team so that everyone is aware of the monitoring activities and updated during the implementation of the trial. So the benefits of the risk-based management approach is that you're demonstrating adherence to good clinical practice and regulatory guidelines. You're focusing on critical data and processes. It's a more efficient use of resources to mitigate those higher level risks. And it offers a variety of activities to incorporate into the monitoring plan and not just relying on that source data verification. It also allows for targeted approach based on the quality tolerance limits that are set and the key risk indicators that are used. So the next step for public health sciences, we want to continue to promote awareness and education for risk-based management and work with research teams to implement some of these techniques so that the research teams are comfortable implementing them and they can see the benefits of the risk-based management approach. And we also want to encourage a more holistic risk-based management practice for monitoring plans moving forward. So I'll leave you with some education sources. The Clinical Trial Transformation Initiative has a good source of quality by design information, has many recommendations and resources for quality. Transcelerate by Pharma has that risk-based monitoring solution, and the Association of Clinical Research Organizations also has an initiative on risk-based quality management. Some references for today's session. And I'm interested how are risk-based monitoring components being used in your research, and if you would want to share any experience you have with the process. And I'll also leave you with our contact information. If you'd like more information on risk-based monitoring, you can reach out to Dr. Valencia-Moulton or myself, or submit a Penn State research request form.
Is that the end of your slides, Kelly?
Yep, that's the end.
So thank you very much for this very nice introduction to clinical trial monitoring. Let's see if our audience has any questions. To our audience, if you have any question, please feel free to post on the Q&A box, or if in case you want to speak, just raise your hand. There's a button, raise hand, then I can allow you to talk. Oh, here there's one question. It's from Terry. Why is it important to solicit ethnicity before race? Kelly?
Yes, I am looking for the guidance document. So the recommendation is that trial participants self-report their race and ethnicity information. And those individuals permitted to designate a multiracial identity. So when the collection of self-reported designations is not feasible, we recommend that the information be requested. Oh, sorry, doesn't really answer the question.
Hi, Kelly. Can you hear me?
Yes.
Yeah, it's no worry. It was interesting to me. My wife is Hispanic. I'm white American, and with our kids, it's always curious that we put down Hispanic ethnicity, and sometimes they put mixed race, and so I'm acutely aware of the difference between ethnicity and race. And what caught my attention early on was you said, well, we always recommend reporting ethnicity before race. Am I making that up, or did you say that earlier? And I that's what I thought I heard, and it was just curious to me. I thought, wow, I thought there must be some reason. I know in my own extended family, there are people who are, their racial thing ranges from white as snow to very, very black. And so it's just kind of interesting to me. There's always, when I do studies, like I'm doing one study now where I'm building a risk model for the black subcohort, for the white subcohort, and the Hispanic subcohort. And because Hispanic is this ethnicity, which is a mix of races, it often comes out somewhere between the black and white, which isn't surprising. But that just caught my attention, and it's not of any importance. But the way you said it, it made me think, wow, there's some intention there. There's some, over time, people have learned collecting this information that it might be helpful or constructive to solicit ethnicity before race. And I was just curious, but I don't want to take up group time if it's not something immediately accessible.
Yeah, in the guidance document, it just says in order to be consistent with OMB and other recommended best practices, the FDA recommends using the 2-question format about race and ethnicity information with ethnicity question preceding the question about race. So I can look a little bit more to see what the rationale is for that. But they do say it's to be consistent, and it's the other recommended best practice.
Sure. Well, thank you. Very nice presentation. And I don't want to take up more of the group time.
Yeah, thank you.
Thank you, Terry.
It's a good question.
Thank you, Terry. That's a very good question. Yeah. We will look into it and get back to you separately with our findings.
I don't want anyone to lose any sleep about this. So
Well, as a Latina, it's my duty now to find the right answer for you. So that
all right, I got your attention. I hooked you.
Yeah.
Now you won't
definitely.
rest. Right?
Yeah, exactly. Yeah. My guess would be that it could be more comprehensive, and it could also be an indicator that somebody of Hispanic heritage most likely could be mixed race, but that's my guess based on my own genetics and what I have learned for looking at my own background. But I guess that's just my guess. We will definitely look into it and get back to you.
I'll bet you're right. It maybe it primes the person to think more like, oh, wow. So ethnicity, me being Hispanic isn't a race. And then they think, maybe I'm sure there's some systematic reason why they do that to make the person think about it, because I think most people, if you're a white person in our culture, you probably just don't even think about these things. Okay, thank you.
Yeah, maybe to reduce some potential bias in answer. There's just another question. I don't know if you are able to see the Q&A box, but let me read it out. Is there a good resource for defining protocol deviations versus exceptions versus violations?
Yeah, I don't think exceptions are a good terminology to use. If you've deviated from the protocol, it's a deviation. If it's a deviation that's going to change the protocol, then the protocol would be amended to not have that occur. So
Mhm.
I don't see exceptions being used very much in terminology. It's primarily deviations is the terminology, and then people can assign more weight to certain deviations if they want and make them violations or major or non-major deviations. But I think a deviation is a deviation.
Yeah, I
Yeah,
totally
and
agree. This, oh,
go ahead.
sorry.
No, and
Yeah,
I just wanted to say that exceptions, as Kelly said, it's not something that we necessarily would like to see to occur. If that's the case, then that should prompt the investigator and the research team to revise their research protocol to make sure that they are not that they are implementing a procedure that is actually going to be that they are going to be able to implement as approved by the IRB.
hey, any other question? If you like to talk, just like Terry, we can do that too. Just raise your hand.
All right, I'll ask one more.
Mhm.
Yeah.
So Paula and Kelly, if we're starting a study and I've, analyzed clinical trials, but I haven't been in one from the start in this formal way, I guess we come to you guys and you guys can guide us through this. You could tell us, okay, this is the risk-based monitoring that you should consider, and maybe with this periodicity of meeting, in other words, you guys could get, if someone wants to start, you guys can guide them through this process.
Correct.
Okay, so you're
Yeah.
educating, but you're also there to guide and consult if needed.
Yes. So, Kelly, do you want me
You can go
to
ahead.
answer? Oh, okay. So, we receive funds by the College of Medicine to provide consultations free of cost to a subgroup of clinical trials. If, for example, the investigators are conducting a clinical trial that is multi-site or is defined as high risk, or it's utilizing a new drug or device, they are referred to us by the IRB and or the research quality assurance offices for clinical trial monitoring services. And those services, except for travel expenses, are covered by the college. So the investigator doesn't have to pay for that consultation and or visits and processes that we might implement for them. On the other hand, if the investigator initiated trial on any of those criteria, we can still provide these services through a fee-for-service structure. So in that case, we will meet with the research team. We will develop a proposal that will include the proposed activities as well as the costs associated with it. And then we can decide how to move forward.
Wonderful. Yeah, it makes perfect sense. It's a super valuable skill and someone has to pay for it. So it sounds like there are different arrangements based on
Mhm.
the nature of the grant, right?
Correct. Yeah, and we have a team currently of Kelly acting as the project leader for clinical trial monitoring services. We also have Rebecca Derbez as one of our clinical trial monitors, and we are in the process of expanding our bandwidth. So we are more than willing to collaborate with investigators across the different campuses. This is one of the efforts that we are implementing in order to increase awareness about these services and develop those relationships in the benefit of Penn State research. That's our main goal.
Do you have like with BERD, right? Anyone can come and get 4 free hours of biostats consultation. Is there something similar with clinical trial monitoring?
No, I mean, yes and no. The source of funding is different because BERD and data management support for REDCap are funded through the CTSI.
Yes.
So that's a different venue. However, we do work closely with the statisticians and with the data managers. So if we identify at any point that you could benefit for any of those services, we are able to connect you with those groups and vice versa. But the source of funding is different, so the way to reach out to us is different. So you could either be referred to us by the IRB or RQA, who recommend to you to utilize clinical trial monitoring services because your clinical trial meets any of the criteria, or you could reach out directly to me or Kelly and we can discuss a proposal with you.
And we
Wonderful.
are
Thank
able to help investigators at the time of grant proposal. So if you want to build our
Yes.
services into your budget, we could work with you to come up with an initial consultation for those and help you to build that into your study.
Correct. This is something that we have done with several groups already, where, for example, the same way that you include in your budget money for data manager or statistician, we can provide the estimate and the budget justification or any boilerplate for the grant proposal, so we can be involved from the very early stages of your effort. And to be honest, the early we can start working with you, the best the outcome is going to be, because it's easier for us to work according to a budget that we customize for your grant than trying to accommodate our services to the funds that you already have or plan for without us being involved early in the process. It gives us more flexibility and it ensures that we have all the components and all the effort in place in order to provide the best quality service to you.
Wonderful. Thank you so much for
Thank you.
super important resource.
Thank you.
We can probably take one more question. Any other question?
And just before we disconnect, I just wanted to offer, Kelly included our contact information. If there were any questions that we were unable to answer, please don't hesitate to reach out to any of us, and we will be happy to talk in more detail with you or answer any questions.
Yeah, sure. I think each participant will get these slides. I think at the end of the slide, you have the contact information. All right, it's 5:01 now. I think it's okay for us to end this seminar. Thank you. Thank you, Kelly and Paula, very much for the excellent talk. And also thanks, Terry, for the nice questions.
Yeah.
All right.
Thank you, Kelly.
Thank you. I hope to see some of our participants in the future seminars. All right. Have a nice day.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye.
Thank you. Bye.
Bye.